Hello to the four of you that have joined us already. Hopefully, we'll get a few more to join us in a bit. Somebody's already left. Was it something I said? Um, but hopefully, this live stream will be helpful to you. My plan is to mostly let your questions kind of guide the discussion here. And if there is a lull in the action, as promised, I took some of your questions that we went over in class last week, and what I thought was a pretty productive little Q&A session during class on Thursday and Friday, and we can sort of fill the time with that. And I'm prepared to go at least 45 minutes or so. Um, but before we start, hopefully everybody's drinking a good hydrating beverage. Perhaps some caffeine if you're staying up late studying. Don't stay up too late. You need a good night's sleep, especially if you're taking my midterm tomorrow. I'm going to go ahead and do some buzz marketing for Trader Joe's. Their sparkling cranberry clementine spring water is very tasty. Highly recommended. So after I hydrate a bit more, if anybody has any questions, I think you know how this works by now. If you would like to add your question to the live chat that you should see on the right side of your screen, you're welcome to do that. Or if you would like to email me, then you can send me an email too. I have my email open and we we can do it that way as well. I'll give you a minute to kind of formulate questions. And in the meantime, while we're still waiting for a few people to come in, I know high school students are usually fashionably late. We usually kind of stall for a few minutes. Let's talk about the test. So, your test will be 80 multiple choice questions. It's going to work out to 3/4 of a point apiece because that part of your grade will count 60%. And then you'll have an open response section where you will need to answer four of the five questions provided for you. There will be one lab practical station focused on the skeletal system. It'll be very, very simple. So if you know basic bones, the joints that they're a part of, and sort of the basic bones and sutures of the skull, you should be good to go there. And I apparently some signals got crossed last week and something got lost in translation. I thought I had explained things the same way for all my classes, but just to clarify, and shout out to Peyton and Shannon for making sure that this information was correct. You do get to use both sides of a 3x5 note card on tomorrow's test. Still no pictures. I have a strict no pictures rule in my tests, but you can put whatever information you think will help you. That being said, there's really no substitute for studying. Those of you who are taking this tomorrow, it might be too late for you. If this is the first time you're thinking about this test, then tomorrow could be a little rough, especially with some of the early stuff. But if you've chipped away, if you've done a little bit each day, and if you've prepared well, then really the note card should just be a reference. When you can't quite remember something, when you have to figure out how something is spelled, something like that. That's the sort of stuff that you should use a note card for. We have our very first question. Shout out to Sam for putting a question out there. Now, I did. I gotta be honest. My proofreading skills could be improved, and I did put some questions related to bone physiology. We did talk about bone physiology briefly. And the question that Sam asked, which some of you can probably see in the live chat box over there, is what is the significance of an epiphyseal plate in a long bone? We did talk about this briefly in class. And in a long bone, and the femur is sort of the typical example that we always talk about, there is the diaphysis, which is the long shaft of the bone, and then the the epiphyses, one is an epiphysis, two are epiphyses. You have epiphyses at either end. So the ends of the bone are called epiphyses. Now, the space between the shaft in the middle and the two ends is called the epiphyseal plate. And that is a band of cartilage, which is where bone is laid down. You have these cells called osteoblasts. That came up last week rather controversially. And those cells sort of change that cartilage to bone. As time goes on, those cells divide and lengthen, and that's how you grow. That's how your long bones actually expand. So you go from having very, very sort of short and thin bones when you're very little to having the hopefully fairly long and fairly thick bones that you have now to support increased body weight, increased muscle mass, things like that. Especially during puberty, muscle mass increases. And so hopefully the density and the thickness of bones will increase along with their length. So when you have a growth spurt, those cartilage cells are dividing and lengthening really quickly. And it's only when that epiphyseal plate changes from cartilage to bone somewhere in your mid 20s that you stop growing, theoretically. A lot of you probably won't grow anymore. Sorry to spoil it for you, but because that's a pretty complicated process mediated by a lot of different hormones. But the big difference between, say, an X-ray of your femur and my femur, and I just had an X-ray of my femur done when I had my knee stuff. Is that the end of my femur, there will just be a very thin line. That's where the epiphyseal plate once was. You would have a slightly thicker band of cartilage in your bones that would show that your bones are still capable of growth. So when you break a bone right at the end, the doctor might be concerned that you broke a growth plate, and that's what the epiphyseal plate is. So maybe that was a little bit rambly, especially for something that's not really going to be focused on the test, but it's a good question. Thanks for starting us off, Sam. So, what else do we have? I'm fully prepared to pull something out of the old grab bag here. I'm guessing it's probably going to be around something about biochem. But she also asked, Sam, you're on a roll tonight. What are some examples of irregular bones? Irregular bones. So we talked about long bones, which are most of the bones of the limbs. We talked about short bones, which are all the bones of the hands and the feet. The carpals, metacarpals, and phalanges on your hand. The tarsals, metatarsals, and phalanges of your foot. I'm not going to hold up my foot to show you that. You know what a foot looks like. Nobody needs that. And we also have, what's the other one? Flat bones, like the sternum, like a lot of the bones in your skull. And irregular bones are kind of just what they sound like. They're very irregularly shaped. They have all sorts of funky nooks and crannies and stuff sticking out of them. The mandible is an example of an irregular bone. The sphenoid and the ethmoid and the skull are examples of irregular bones. And the one that we've probably looked at the most is the vertebrae. The vertebrae are sort of literally a textbook example of irregular bones because they have all sorts of processes. They have parts sticking out. They have extra openings, those transverse foramina on the cervical vertebrae, all kinds of funky stuff going on. So anytime you see a bone that has a lot of irregular features, not to use the word in the definition, but that's what an irregular bone is all about. So hopefully that helps. What else we got? All right. So while we wait for another question, let's pull something out of the old grab bag. I really wish I had like a guest host here to help me with this, but I don't know if I'm quite comfortable inviting you guys over to my house to co-host this live stream with me. We're not quite there yet. Okay. We're gonna put that one aside. That might be a little too specific. Let's see. Will we need to know the bones in the hands and feet? Very good question, Ian. I would expect you to know where the phalanges are in the fingers and toes. I would expect you to know that the thumb and the big toe have 2 phalanges while the others have 3. I would expect you to recognize the names of the tarsals. So, like, if I asked you which of these bones is a tarsal, then I would expect you to be able to pick out, for example, the calcaneus among that. You won't need to label anything or do anything like that with respect to the hands and feet. We're not, like I said, we've already gotten very specific with the skeletal system, so the questions you're going to have to answer here are going to be quite general. So, and now we're coming with the questions fast and furious. Good stuff, guys. And we got 13 people. Lucky number 13. What is the chemical composition of an enzyme? Well, the long answer is that it really depends because obviously there are many different enzymes that look very different from each other. But long story short, enzymes are proteins. They're proteins that speed up chemical reactions. We say that they catalyze those chemical reactions, and they do so by binding to a substrate. That's the one molecule that they interact with, and decreasing the activation energy of that reaction. That is the amount of energy that needs to be put in. Because enzymes are proteins, they're very sensitive to changes in temperature and pH. One of my favorite go-to questions in that biochem unit was let's say there's an enzyme that breaks down carbohydrates in the mouth. Salivary amylase is the one that we talked about quite a bit. That enzyme will not be able to break down anything else, first of all, because it is very specific to starch. So it won't be able to break down lipids or nucleic acids or anything like that. And it also will only be able to work in the pH of the mouth. Any lower or higher pH may denature that enzyme, may cause it to lose its 3-dimensional structure. And if it loses its structure, it loses its function. One of the things we've talked about practically since day one with almost any molecule, but proteins especially, is that structure follows function. So if a particular molecule loses its structure, then it's not able to perform its function as well, if at all. And in the case of many enzymes, once an enzyme is denatured, it can't really regain its function. So hopefully that helps. Okay, so what else do we got? Feel free, you don't have to wait until I finish answering a question to put in another question. We're not really being overly formal here. I'm sitting here in a hoodie after getting back from the gym. So feel free to just throw questions out and I'll answer them as they come. Ashley asked, does the midterm have a lot of questions concerning the central nervous system and peripheral nervous system? The basics of the nervous system that we've already done are certainly fair game. When we're talking about a lot of what we've talked about already deals with mostly the peripheral nervous system, but really we've talked about how any neuron, whether it's in the brain or spinal cord or outside, sends signals through action potentials. So I would know about the roles of sodium and potassium and that. I would know a little bit about the steps in an action potential. I would definitely know the parts of the neuron and why they're important. Know a little bit about why myelin's important and the nodes of Ranvier. We did just assess on it, but it's part of first semester, and so I feel like it's fair game. I I don't know if that's a satisfactory answer, but if you want me to expand on that, I'm happy to do that. In terms of bone marrow, really, you need to know that bone marrow is found in the center of bones and that it makes blood cells. In fact, all of the blood cells in our body. That's why certain diseases like cancers that affect the bone marrow, blood cancers, often affect the bone marrow and affect one's ability to make red blood cells, white blood cells, platelets, all sorts of things. My wife, not to brag, uh, tries to donate platelets as much as she can. She used to donate blood all the time, but she realized that she could donate platelets like every 2 weeks. She could donate like up to 24 times a year, so she could do it twice a month. And she donates a bunch of platelets, and a lot of those platelets are given to cancer patients, especially patients are having who have cancers that affect the bone marrow because they can't properly make platelets, and therefore if they bleed, if they get a bruise, if something happens, then their blood might not clot and they might have further health problems. So it's actually really important for platelet supplies to be constantly topped off because they also don't last as long as whole blood does. You can keep a unit of whole blood for a couple weeks. You can only keep platelets for a couple days. So like when they get them, they have to use them right away. So like even on like my wife went and donated platelets on Christmas Day because they need platelets like literally every day. Um, so the bone marrow is a pretty important thing. And if there's something that affects it, that could be a problem. We'll talk a little bit more about the bone marrow when we get into the cardiovascular system later in the year. But for now, if you know what it is and what it does, you're going to be fine. Okay, no questions on my email just yet. Um, so let's go into the grab bag. See what we got. I bet it's a question about biochem. If the patterns hold. Okay. This actually is one that I didn't get to. So I'm thinking we'll talk about this. What do we need to know about the organs? So the stuff we did about organs was way back in the first unit, way back in September, when we. Oh, hey. And Ava's question is definitely related to that. So you should definitely know the organ systems and which organs belong to them. I would actually like to step out for a moment because I've been hydrating very well. And when I get back, I'll kind of do a general overview of all the organs and organ systems, and I think that'll be a good use of our time. So sit tight for just a minute. If anybody else comes in, reassure them that I'm not. I haven't left. You'll be staring at my back wall, and I'll be right back. Okay, so sorry to keep you waiting, but let's talk organs and organ systems. So I will expect you to know sort of general functions of some of the major organs that we've talked about. For instance, we talked a bit about the lungs and their role in not only not bringing in oxygen and expelling carbon dioxide, but in regulating pH. We talked about the role of the kidneys in regulating water content and regulating pH. We've talked about a little bit about the heart and the blood vessels and their role in controlling blood pressure. So, where they've come up, I might expect you to know a few more specifics. But in general, I would expect you to have a good general knowledge of all the organs and the organ systems that we've talked about. So, um, let's start on the outside. Right here, we have the integumentary system. We're talking skin, hair, nails, all of the sort of outer protective layers of the body. and that's pretty much its function, to protect the inside of the body rather from all of the things that could cause physical damage, could cause infection, things like that. Then if we go inside, if we want to move, then we depend on two systems: the skeletal system, which we've already talked about a whole lot, and the muscular system. Skeletal muscles in particular attach to those bones. In fact, most of the little processes and other things that stuck out of bones are attachment sites for muscles of one type or another, whether it's, muscles in the skull that attach to the mandible and help us move the jaw up and down, or whether it's the, quadriceps muscles that all insert right on the top of the tibia at the tibial tuberosity. All of those little bumps and nooks and crannies and processes are usually places where muscles attach. So those two are responsible for movement. Then to get those cells and all of the other cells the oxygen and nutrients that they need, you need the cardiovascular system. So the heart and the blood vessels. The heart pumps, the blood vessels use their sort of smooth muscles to push that blood and all of the nutrients and oxygen that it contains through the body. Um, but you can't get oxygen into the cardiovascular system without help from the respiratory system. So the lungs, the trachea, the mouth, the nose, all of those could be considered part of the respiratory system as well. All of this, of course, speaking of the nose, is governed by the nervous system. The nervous system consists of the brain and the spinal cord and all those peripheral nerves, as well as the sensory receptors on the surface of your skin. So you could consider the skin kind of an organ of the nervous system as well as all of the sense organs. So the nose, there's a lot of nervous tissue in your nose, in your eyes, in your ears. So the fact that you're able to listen to me and see my shiny face right now, I'm really bothered by all this shine on my forehead here. I got to get better lighting. That is a consequence of the nervous system, as well as your ability to react to stimuli and to control your environment. Now, another system that really helps with that is the endocrine system. Both of them, the nervous and the endocrine, communicate using chemicals. In the nervous system, we're talking neurotransmitters, things like dopamine that make you feel real good, things like serotonin that help regulate your moods. And there was one that we talked about in at least one of my classes called acetylcholine that basically it communicates with skeletal muscles to make them move. So every time I want to do something like this, I think this is going to be the lasting image from this live stream. I need to send acetylcholine to the muscles of my arms and my hands to make myself dance. I'm so sorry. So we've talked nervous, and the endocrine system is very similar in that it plays a really important role in homeostasis. For example, a lot of feedback loops do involve the endocrine system. When we get into the endocrine system in a few weeks, you'll see what I mean. And it communicates through hormones that are secreted by glands. And there are lots and lots of glands, the pituitary being really important, the pineal gland that secretes melatonin that helps you stay awake and go to sleep when it's appropriate. The thyroid that regulates metabolism, that's the one that doesn't work so well in my body. The parathyroid that helps regulate calcium, the adrenal glands, the pancreas, you name it. Again, we're getting way deeper into the endocrine system in a few weeks, but for now, just kind of know a few examples of organs that come from that system. And of course, the digestive system. So, the digestive system is a really important system in that it allows us to break down the food that we're taking in to derive energy from it. Energy in the form of carbohydrates, which are broken down and converted to energy in the form of ATP. And it also provides raw materials to build things. We can break down the proteins that we took in through our food and break them down into amino acids, which can then be made into new proteins. That's why after a hard workout, when you've done damage to your muscles, it's really important to get protein right away in order to help those muscles recover. Um, and let's see, that was kind of an aside. We also have the urinary system or the excretory system. That's the kidneys and all the associated organs. And of course, we have the reproductive system, which is responsible for making sure our species continues on this earth. In females, we have the ovaries, which store the eggs, the uterus, which develops the eggs, the vagina, which is sort of the reproductive opening. It is the reproductive opening, not sort of. And in males, you have the penis, which is the male reproductive organ, as well as all of the associated structures that help to produce healthy sperm, the testes, and a lot of the other structures that help build healthy sperm that can fertilize an egg, and the miracle of life. So, can you talk about homeostasis? I would love to talk about homeostasis. You know, I love homeostasis. I also love this seltzer. Seriously, guys, this is life changing stuff. Go get it. Not right now. Trader Joe's probably closes very soon. But so homeostasis. The mechanism by which homeostasis works is called negative feedback, as you know, and it operates in such a way as to reverse the original stimulus. And the stimulus would be, in this case, a change in a variable, and restore that variable to normal levels. You hear a lot about the idea of a set point for a given variable. There's a lot of theories out there about how we have set points for weights and things like that. There's sort of normal levels of blood pressure that doctors would expect you to have. All of those are regulated by negative feedback. Blood pressure is a great example. It's one we talked about way back at the beginning of the year, and we brought up a little bit last week, at least in some of my classes. Sometimes I don't remember exactly what examples I use, but the regulation of blood pressure is very tightly controlled. So let's say that you lose some blood. Let's say you donate some blood. It's a very good thing to do. You should go do it. And so you're down a pint of blood. When you give just one unit of blood, they take everything. They take what they call whole blood. So they're taking the plasma, which makes up a little more than half of your blood. They're also taking all the cells, the red blood cells, the white blood cells, the platelets. So because they can use it all. So they're gonna take that whole blood, and you're gonna be down some fluid. If you're down some fluid, your blood pressure goes down. So the way that you detect that is through sensors that are actually in your major blood vessels. So your aorta, your carotid artery, things like that, they all have these sensors that sort of detect when blood pressure is lower than it should be. Now, the regulation of that, so there's the sensor or a receptor. That's the first component in the feedback loop. That's going to send information into the central nervous system through a sensory or afferent neuron, and it's going to land somewhere in the CNS. This goes for most impulses. Sometimes the central nervous system doesn't need to be involved, but most of the time it's going to be sent to what we call an integrator. And that is some component of the central nervous system. And in this case, we're talking about a structure called the medulla oblongata, right at the base of your brain. And the medulla oblongata has a few centers that control sort of automatic functions, things that you don't have to think about. There's a center there for heart rate. There's a center there for respiration to control the rate of breathing. And there's also a center that controls blood pressure. So that receives the information, realizes that blood pressure is too low, and it sends a signal out to an effector. And the effector is what actually brings about a change. In the case of negative feedback, which is the mechanism of homeostasis, again, that change is going to, that the effector brings about, is going to reverse the original stimulus. So blood pressure goes too low, it's going to bring it back up. And the effectors in this case, usually they're muscles or glands, sometimes they can be other things. And the, God, what was I thinking? It is 8:33 p.m. and I have been going all day. But the effectors, in this case, are blood vessels, which can constrict. They can reduce their diameter and get really tight. So that increases blood pressure. And the heart, which can beat faster and with more force to generate more pressure within the heart, which then translates to all the other blood vessels. And in doing all those things, the blood pressure is brought back to normal. I would be familiar with positive feedback. Ava just asked a related question, so I figure I'll address this now. Positive feedback is something that happens when you want something to happen really quickly. The classic example is the clotting of blood. When you, and that's why you need platelets, because platelets, they rush to the site of the injury and they form what's called a platelet plug. At the same time, they're releasing a chemical that summons more platelets to the site of an injury and like basically allows that platelet plug to grow. And it keeps happening. It keeps sort of moving that stimulus in the same direction, more platelets, more platelets, more platelets, until that plug has completely sealed that injury. Again, if you have enough platelets and if they're releasing these chemicals properly, then that will very quickly stop bleeding in most cases. Childbirth is another great example. It starts with contractions of the cervix, and oxytocin is the chemical that is released to stimulate those contractions. And when those contractions start, it triggers the release of more oxytocin, which triggers more and more powerful contractions, which triggers the release of more oxytocin, and so on. So I would be familiar with it. I think it's reasonable to ask that based on what we've talked about. So I'm going to back up a couple questions here. I need to take a sip of water. This is a lot of talking. Okay. Is there a lot of biochemistry questions on the midterm? Sure. I expect you to know about carbohydrates, proteins, and lipids, what they're made of, what their building blocks are, and what they're used for. I think that's basic information that you should know as a student of anatomy. You should definitely know how enzymes work. We talked about that quite a bit last week. You should definitely know about some of the properties of water that make it really special. It's high heat of vaporization that helps us cool the body down by sweating. It's high heat capacity that helps to control the temperature in the body because water requires a lot of input of heat to raise its temperature. And you have to remove a lot of heat to lower its temperature as well. So that allows our temperature to stay consistent. And you should also know a little bit about acids and bases, pH levels. So that's a great segue into Sam's question. Thank you for providing that segue, Sam, because I would love to talk about pH levels. So, as you know, anything with a pH below 7 is an acid. Those things tend to release hydrogen ions when they dissolve in solution, when they dissolve in water, when they form a solution. HCl, hydrochloric acid, is the one that we find most in our body. That is what makes up most of stomach acid. That's what accounts for the low pH in the stomach. And the anything above above 7 would be considered a base. Bases technically release hydroxide ions, that is OH minus, when they dissolve in water. Most of the base that we're going to find in our body is in the form of the bicarbonate ion, and that's HCO3 with a -1 charge. Um, it's very similar in its sort of chemical behavior, but that is what allows us not only to keep the pH high enough in certain areas like the mouth and the small intestines where a higher pH is expected, but it also allows us to it acts as a buffer, and that buffer helps to control large scale changes in pH. You may remember a demonstration from way back in the day where we had a buffer system that we could add large amounts of acid and base and it wouldn't change the pH that much. That's a really important thing in the body because, as we discussed earlier, enzymes in particular and molecules in general are really sensitive to changes in pH. If the pH goes up too much, which we would call alkalosis, or if the pH goes down too much, which we would call acidosis, then the enzymes that we depend upon to speed up or in a lot of cases actually bring about chemical reactions, like they don't happen if enzymes aren't present, they would be denatured, they would lose their three-dimensional structure, and we wouldn't be able to use them anymore. So clearly, it's really important to control pH. We talked briefly about the role of the kidneys in doing that, in keeping more hydrogen if the pH needs to go lower, or keeping more bicarbonate if the pH needs to go higher. Frankly, I would be more concerned with the role of the lungs in regulating pH. And it's going to do that by controlling the amount of pH of CO2. We know that carbon dioxide, when it dissolves in water, forms an acid. So, the more CO2 that you retain in your lungs and in your bloodstream, the lower your pH will be. Someone with lung issues, like my parents, both have pretty crappy lungs. This is what 35 years of smoking will do to you. And there's been some evidence recently that vaping does the same thing. So don't vape, kids. Um, and when your lungs start to deteriorate through things like emphysema and things like that, then it becomes more difficult to expel CO2. And so you're in a nearly constant state of acidosis. And the way to get rid of that for most people, an adaptation that you would have would be to get rid of CO2 by hyperventilating. If you needed to retain carbon dioxide to keep the pH down, then you could breathe into a paper bag. That would simulate a process called rebreathing, where you're literally breathing the air that you just breathed out. Or you could slow down your rate of breathing, or in some cases, even stop it for a period of time. And we're talking 10 seconds in order to retain some of that CO2 and keep the pH down. So hopefully that covered it. Osteoblast came up in the review last week. That heads up review that we did, I forgot to remove the word osteoblasts. We didn't get into that kind of detail about bone physiology. Osteoblasts are cells that sort of churn out bone matrix. They build bone. They're really important in bone growth and in bone remodeling when you have to retain bone mass. And so we're not going to worry about the function of osteoblasts for now. We didn't get as much into bone phys as I wanted to. Most of the bones that we studied were endochondral bones. So your femur, your humerus, all of the long bones are endochondral bones, which means they develop from cartilage models. Intramembranous bones are kind of exactly what they sound like. Intra means within, membranous refers to membranes or layers of connective tissue, and that's pretty much what happens. So a lot of your flat bones, especially the bones of the cranium, for example. So the frontal bone formed between two little layers of connective tissue, and there were osteoblasts. There they are again in between that just laid down bone, kind of like a filling in a sandwich. So if you think of like a PB and J, the two layers of connective tissue are the bread, and the bone is sort of the filling in between. That's a basic explanation. I might ask you to be familiar with those words, but I'm not certainly going to ask you too much detail about them. So Jenna has asked a couple questions here. So let me backtrack here. First one, she asked, what do we need to know about the needs and requirements of organisms? So, I would say in terms of, I don't know that a lot of them are going to come up explicitly in the midterm itself. Certainly, organisms need to be able to respond to stimuli. They need to be able to control their internal environment through homeostasis. They need to be able to maintain boundaries. That's the job of the skin, certainly, as well as cell membranes that help to protect cells from what's outside. They need to be able to receive nutrition. In our case, we eat and we break down that food to produce nutrients. They need to be able to grow and develop. But I feel like a lot of that is going to come up in the context of other things. I don't know that I would necessarily ask you a specific question about those needs and requirements. I think that we talked about those a lot in the beginning, and we've kind of kept, hopefully, I've kept sort of providing evidence that those are, in fact, things that living things, specifically humans, need. Um, I just thought of how we spend a lot of the year sort of proving that those are actually things that we need and getting into detail about how we fulfill those needs. Good stuff. So I would say you already know more than you think about the needs and requirements of organisms. I'm thinking that I wouldn't spend a lot of time like memorizing those needs and requirements. I would say that you're gonna use them in the context of the other things that we've studied. In terms of reflex arcs, um, a reflex arc is pretty much what we've talked about in terms of reflexes. So just to review, we didn't get to actually carry out a really nice reflex lab that we usually do. I think we might actually do that like next week to kind of continue with our nervous system units, just because it's a fun lab to start us off after midterms. But let's say that you're testing the knee jerk reflex. So in the knee jerk reflex, sorry about my leg here, but you're going to hit the patellar tendon, which sits right here. And what that's going to do, so I'll kind of walk you through the reflex. And the withdrawal reflex that we've talked about in class is another example of a reflex arc. It's just a term that we use to describe sort of the path that the signal follows during a reflex. And so a stretch reflex, like the knee jerk reflex here, that would start with that connective tissue, which has stretch receptors. So when that tissue gets stretched, like when you hit it with a hammer, or when you straighten your leg to sort of walk, it's late, guys. When you straighten your leg to walk, that also triggers that stretch reflex. And so what happens is that the stretch receptor sends a message through a sensory neuron up to the spinal cord. Remember, a lot of reflexes are simple enough that you don't need the brain involved. The spinal cord can handle its business on its own. So you send an impulse through a sensory neuron all the way up to the spinal cord. The spinal cord has some interneurons. Remember, those are the ones that sort of do the processing in the central nervous system a lot of the time. And that interneuron will send a signal directly out to the quadriceps muscles right on the front of your leg. They will contract and you'll kick out. And you won't have to think about any of that. That's really almost an autonomic thing. And you could sort of control that and almost suppress that reflex in a way. That's why doctors need you to relax when they're testing your reflexes, because if you don't, you might not be able to see a reflex that you would normally have. But in terms of the term arc, that really just describes the pathway that we follow from that sensory receptor into the spinal cord all the way back out to the muscle, which then contracts. So it's just one way to describe that reflex. Hopefully that helps. So we are at the 46-minute mark, guys. Time flies when you're having fun. So we've got about, by my watch, we've got about 13 more minutes or 12 more minutes. It just hit 8:48 on my clock. So I am happy to help with anything in the next 12 minutes. You're welcome. And if there are no more questions for now, I can pull something else out of the grab bag here. Let's see. This is exciting. Biochem, function of organs. I feel like we've covered those pretty well. Certainly, if there's a specific aspect of biochem we want to discuss, we can talk about that. But first, Sam has a question about action potentials. So, action potentials, as you remember from last week and the week before, are the way that nerve impulses are sent from a neuron to some other cell. And so, and a given neuron receives a signal through its dendrites. The dendrites are the ones that receive the signals. And let's say, for example, that we're receiving an impulse from a sensory receptor after we put our hand on a hot stove. So that signal comes through the dendrites of this neuron, then through the cell body, and it arrives at the axon hillock. That is the first part of the axon right where it meets with the cell body. So we might call it the proximal part of the axon, right at the beginning. The other name that we use for that is the trigger zone, which I think is a super cool name for anything. And it's at the trigger zone that we have all of these sodium ion channels. You remember that sodium is key in initiating an action potential. The sodium ion channels open up as soon as a strong enough stimulus is received, what we call a threshold stimulus. And those ion channels will allow sodium to diffuse into the membrane. So it's the influx of sodium into the membrane of the neuron that changes the electrical potential across the membrane. You remember it starts at rest being more negative on the inside, and in order to generate an action potential, it needs to start to be more positive on the inside. And sodium does that very well because it's positively charged. So sodium goes in, it depolarizes the membrane, makes it less negative, more positive, if you will, and sends an action potential. So that's how you start it. Now, you also have to reset the neuron, or that neuron could potentially stay in that sort of excited state. That's how cramps happen, because you can't relax your neurons, the neurons that connect to muscles specifically. So that's the job of potassium. You remember that we start with a lot of potassium inside. So right after the sodium ion channels allow sodium to diffuse into the membrane, potassium ion channels are going to open up, and then potassium can diffuse out of the membrane. It can diffuse out of the neuron. It starts to bring that potential, that difference in charge, back down to make it more negative on the inside, because we're losing positive charges now. And eventually we get back down to the resting potential. Usually more potassium flows out than sodium flows in. So we go a little more negative. We experience what's called hyperpolarization. But luckily for us, we have the sodium potassium pump, which is a protein that sits on the membrane that requires energy to pump, literally, sodium back out of the neuron and potassium back into the neuron. So at the end of this whole process, we restore that resting potential of negative 70 millivolts, which means that the inside is slightly more negative than the outside. And we restore that difference in concentration where we have a whole lot of sodium on the outside, and we keep it that way with the use of the sodium potassium pump. And we have a whole lot of potassium on the inside for the same reason. So hopefully that helps review action potentials. Um, Let me just make sure that was not a question related to this. So the difference between tarsals and metatarsals is very simple. The tarsals are the 7 bones sort of on the back of your foot. We have the calcaneus, which is the heel bone, the talus, which sits on your ankle and joins up with the tibia and the fibula, and a whole bunch of others that make up your sort of the rear foot, and sort of goes up to kind of the front of your arch. Now, in front of those tarsals, out toward the toes, but not in the toes yet, are what we would call the midfoot, would represent the metatarsals. They're numbered 1 through 5, 1 starting with the big toe and 5 with the pinky toe. Then we have all the phalanges on the toes, 2 in the big toe, 3 in the others. And that's a good summary of foot bones. So any other questions? Looks like we got about 6 minutes to go. My clock just struck 8:54. We're going hard tonight. Okay, so I'll go ahead and pull one more question out of here. Maybe we'll make this the last one, unless anybody has any lingering questions. It's been a good discussion tonight. Thanks for helping out. What do enzymes do? We already talked about that. And what do mono and polysaccharides do? So, of course, that talks about the different types of carbohydrates. We have monosaccharides, which are the single sugars, small molecules, relatively small, like glucose, that are used for quick energy. So, a lot of times when you're in a race, when you're doing some sort of endurance event, you're going to take in a lot of sugar, because sugar can be used right away. When you run out of sugar, you need to use other sources of energy, and you want to try to avoid that as long as you can. So you keep topping off that supply of sugar throughout that event. I would usually, in my marathoning days, I would take in probably about every 4 miles, every 4 or 5 miles or so. So every like 30 to 45 minutes, I would take in like 100 calories of carbohydrates, and that would pretty much tide me over, so I wouldn't hit the wall later on. When you run out of those monosaccharides, the next source of energy that you have is going to be polysaccharides, and those are the ones that are stored up. Plants store them up as starch, and then we eat them. So your breads and your beans and things like that have a good amount of starch in them. We store them up as glycogen, which is just a big long chain of glucose molecules that allow us to that we can break down to glucose to be able to use it for quick energy. It takes a little more time, takes a little more oxygen, but we can use it pretty quickly. When we run out of all that stuff, we're going to use lipids. And lipids, gram for gram, because they're so big and they have so many bonds that store up energy, they can give us twice as much energy per gram as carbohydrates. But they also, because there are all of those bonds, they require a lot more oxygen to break down. And so it's a little harder for us to access those and puts more stress on the cardiovascular system. So we prefer to break down carbohydrates if we can. Brief overview on the bones in the hand. Again, we're not getting into a ton of detail with really anything, but basically we got carpals in the wrist, 8 carpals. You don't really need to know them. We got metacarpals starting with the thumb, well, under the thumb, I should say. So the palm of your hand is going to be your metacarpals. So 1, 2, 3, 4, 5. And then you got your phalanges here. Um, if you make a bear claw, you can see all the phalanges. We got 3 in all the other fingers, 2 in the thumb. That's a quick overview of the bones of the hand. And again, if you're I wouldn't get bogged down too much on details in the skeletal system. We've already gone into painstaking detail on those bones, so we're not going to go too deep with that. I can guarantee you will not see a hand on that lab practical question. So hopefully that sets your mind at ease a little bit. If there are any last minute questions, we got about 3 minutes to go, and otherwise we might call it a night. And those of you who have this exam tomorrow morning, um, well, it's crunch time. So hopefully you've been studying throughout. Hopefully you've got a nice note card that's going to help jog your memory if you need it. And otherwise, the rest is up to you. You got everything you need. And you should be proud of the work you put in. So the rest, I guess, we'll find out tomorrow. So if there are no other questions, it looks like people are starting to leave and go about their business. So I'm going to do the same. I've barely seen my wife today, so I'm going to go say hi to her. And I will see you all in the morning. Thanks for watching, guys.